Hello everyone, my name is Madison Eckard. I use she, they pronouns, and I'm a first-year student in biochemistry. My passion for science came from a group of YouTubers who call themselves science education creators. The stimulating visuals, clean, scripted explanations, and digestible content left my curiosity constantly hungering for more. Inspired by those videos, I've prepared this talk, which is more of a performed YouTube video than a rigorous research presentation, but I'd like to believe it is the best of both worlds. So with that context, I hope you enjoy learning how growing human lungs helps us fight pandemics. Over the past year, I have had the pleasure of working in Dr. David Blake's lab on his Lung on a Chip project. This is a form of research that seeks to mimic human organs using a mix of technology and living cells. We have been using lung on a chip to see how lung cells react to hypoxia, or lack of oxygen. The usual culprits of taking away our cells' oxygen privileges include tumors, viral and bacterial infections, and most lung diseases. By studying cellular responses to hypoxia, we learn a lot about how our lungs work and discover new pathways for drugs and medical treatments. However, we will need some way to test said drugs and medical treatments before doling them out to patients. Typically, after rigorous preclinical testing on animals and microorganisms, drugs undergo human trials through the FDA to judge a treatment's effectiveness and safety. 90% of drug trials that enter clinical trials fail, but why is that number so high? Part of the reason is the ineffectiveness of animal testing due to slight differences in physiology. This is where lung on a chip enters the stage, promising more effective drug trials and avoiding the treacherous obstacles of animal and human testing. Today, we will explore why lung on a chip looks to replace human and animal testing, how viruses infect our lungs and cause hypoxia, and how my research connects these ideas to better understand what is going on inside of our lungs. To figure out why lung on a chip will look to replace human and animal testing, we have to understand the pros and cons of each respectively. Much like food tasters who sip their king's wine to test for poison, we test our drugs on a small number of humans before doing larger trials. At its best, human testing can save millions of lives by significantly decreasing the risk of widely administering dangerous drugs. But at its worst, it can lead to some of the most egregious ethical atrocities known to science. Human testing has and will continue to save millions of lives and help lead to many of the wonders of modern medicine. But I'm not sure I need to convince anyone that human testing should be avoided whenever possible. So how then do we make sure drugs won't just kill or injure the first people we test them on? The accepted solution is to look towards animals. Animal testing is a preferable alternative to human testing and is what we use today as our main form of conducting early drug trials and scientific experiments. Using animals as human models is an idea that has become incredibly popular since Darwin's Origin of Species was published. Humans are descended from the same evolutionary ancestry that share many similarities in our organ structures, so it follows that drugs would have similar effects on them as they would on us. This has been awesome news for our species, because now, instead of having to test risky new treatments on humans, we can test it on animals to add a layer of precaution during research. However, animals also share the wonders of having a complex nervous system. Along the same line of reasoning, we could suppose that they experience pain and suffering in similar ways as us. Animals are truly intelligent and capable organisms, and seemingly experience many of the perceptual jewels that we value so highly in ourselves. This growing understanding of the sentience of animals suggests that we should avoid animal testing whenever possible, an ideal held and enforced by all of our scientific institutions today. However, many scientists question the justification for using animals in scientific testing, as they seem to have a lot less predictive value than we originally believed. We now know of many important differences between human and mouse cells, such as different types of cells, the type of cell distributions, as well as their size and shapes. A drug's effect is very much dependent on what cell it interacts with. So it now makes a little bit more sense why 90% of drug trials fail following preclinical animal testing. If animal testing is not so effective, do we just go back to testing on humans to stop harming animals? Well, no, but the answer isn't so black and white. There are alternative models other than animals that scientists today are actively developing, such as computer simulations and testing on cells. Biologists have been using these in vitro tests on extracted living cells since the inception of cellular theory. The issue with these models is that our lungs are not just one uniform type of cell that is copy-pasted over and over again. In reality, they're an ever-shifting mosaic of many different cell types, bacteria, and everything we breathe in and out. With this context, in vitro testing doesn't seem like an effective alternative to animal testing, but we haven't gotten to our star of the show yet. Lung-on-a-chip models are the next evolution of in vitro testing. With these chips, we try to recreate the biochemical environment inside of our body. For example, we might introduce a cyclic vacuum to try and simulate the mechanical stress of breathing in and out, something that could affect how a drug interacts with your body. As these models become more accurate and complex, we can begin to shift away from animal testing and towards more developed models. The lung-on-a-chip model we use in Dr. Blake's lab attempts to mimic a part of our lungs called the alveoli. These alveolar sacs are where a lot of the magic of the lungs occurs. Each of our millions of alveoli allow the exchange of oxygen and carbon dioxide in and out of our capillaries, which connect to the rest of our bloodstream and body. To get a better idea of how exactly our model works, we have to understand how alveoli work, which we will do so through an iconic episode of the Magic School Bus. Oop, there we go. No, it didn't. I love technology when it works. Okay, well, I guess we don't get the clip, but we get to just start. So, in the episode, Inside Ralphie, Miss Frizzle shrinks her students in the titular Magic School Bus to figure out why Ralphie feels sick. They travel through his body, watching the epic battle between his immune system and viruses unfold. Unfortunately, I do not have the budget to shrink all of us inside of a magical flying school bus, but luckily we have modern technology to recreate this visualization. This all begins with one lucky viral particle that finds its way into our nose or lungs. For the sake of brevity, let's name our viral particle, Victor the virus. Victor finds himself hurtling through our respiratory tracts into an alveolar sac. Undeterred by the thick mucus that coats these alveolar cells, Victor attaches himself to the cell membrane, gains access to the inside. There, he will take control of the protein-building machinery throughout the cell and force them to produce more victi. And yes, that is the plural of Victor. As the newly minted victi leave the cell, they face another barrier to overcome: the basement membrane. This is the space between the alveoli cells and the capillaries, full of a wide variety of proteins and cellular components. This dense jungle of proteins prevents blood from entering the alveolar sacs and provides a stable, flexible structure for the alveoli. To mimic the basement membrane in our lung-on-a-chip model, we use a silicon wafer with holes that are chemically etched through it. This lets us avoid building the basement membrane from scratch, while also providing a convenient, adherent surface for cells to grow on top of. The basement membrane is not where our defenses stop, though. As Victor's original cell was destroyed, it disrupted the local oxygen supply to surrounding cells. As hypoxia begins to set in, the cells struggle to keep themselves alive, which will trigger many different reactions to protect our body. Beyond triggering many complex immune system responses, they will also begin producing more of the proteins essential to the basement membrane, in hopes of keeping our body's oxygen supply intact. But in great numbers comes great strength, and the victi manage to break through the basement membrane, gaining access to your bloodstream and thus the rest of your body. Victor has earned his victory in this biological war zone. This explanation is a dramatic oversimplification of what actually occurs during viral infection, but we saw how viruses can cut cells off from oxygen, eliciting several responses. One of those responses is the one we are studying, which is the production of proteins for the basement membrane in response to hypoxia. We saw as cells were growing with 5% oxygen, As compared to the 20% we have in the atmosphere we breathe, they began producing more collagen 4, one of the proteins that make up the basement membrane. These are two images we took of the cells using fluorescence microscopy, staining the collagen 4 green. The darker the green is, the more proteins there are in that specific pixel. Now that we're reassured that it was hypoxia that pushed the alveolar cells to produce more of this protein, we can begin to use lung on a chip to see if viruses have the same effect. This will hopefully give us a better understanding of how viral infections work, providing another piece of the puzzle in the fight against victi in future pandemics. With lung on a chip, animal, and human testing together, we can continue to advance scientific medicine and gain a deeper understanding of how our lungs work. I would briefly like to thank the Intuitive Foundation for providing the grant for me to do this research. I would also like to thank Christine for putting together this wonderful event, Dr. Blake for being a great mentor, Peyton Harrison and Stella Rhodes, with whom I worked with on this project, and Ovia Redling, who helped edit and put together this presentation. I hope you enjoyed learning about the obstacles that scientists face in studying our lungs, how viruses infect our lungs and cause hypoxia, and how our research here at Fort Lewis can illuminate the fascinating cutting edges of biology. The QR code on screen will link to all of the sources referenced in this presentation, as well as a couple of my favorite science education YouTubers whose style I've emulated. Thank you everyone for listening, and have a wonderful day.